Hi, my name is Kate Reinl. I am currently a postdoc at the Center for Limnology at UW-Madison, although I'll soon be switching into a new role as the research coordinator at the Lake Superior National Estuarine Research Reserve located in Superior, Wisconsin. This presentation is going to cover cyanobacterial blooms in Lake Superior, and it covers some of the work that I've done, but also a lot of work that's done by other folks in the area, and I'll talk about those as we move through the presentation. So first, I just want to give a really brief overview of Lake Superior in the context of the other Great Lakes. And I really like this figure because it just really shows the really wide range of trophic states that we see in the Great Lakes, ranging from Lake Erie, which is eutrophic, all the way up to Lake Superior, which for the most part is oligotrophic. However, we're finding in some of the nearshore areas that it might be more leaning towards mesotrophic or, in some cases, possibly even eutrophic. And again, here we can see this stark contrast. We talk about the Great Lakes as this, single system, but really there's a lot of variation from Great Lake to Great Lake. And so on the left here, we have a photo of Lake Erie where you can see, very easily that the water is very green from algal growth. And then on the right, we have Lake Superior where the waters are very clear and pristine, which is commonly associated with Lake Superior waters. And here are some more photos of various locations around Lake Superior where, again, you can see those kind of typical pristine crystal blue waters that we associate with Lake Superior, and what also draws many tourists every year to the Lake Superior watershed, both on the US and on the Canadian side. And while we typically associate Lake Superior with those photos that I showed in the previous slide, Lake Superior also experiences large sediment plumes. Sediment plumes in Lake Superior's western arm are definitely not a new thing, but due to a combination of the region's superficial geology and historical land cover changes, the scale of the plumes following large storm events has been really large, and they've been clearly visible from space and linger for, in some cases, weeks. In addition to the sediment plumes, we are also seeing these cyanobacterial blooms, particularly in the western arm along the south shore, but also in other locations around Lake Superior, including both on the US and on the Canadian shoreline. The map shown on this slide shows a broader spatial overview of past bloom reports, and observations even date back anecdotally to the 1960s and 1980s. However, the recent observations are clustered in the western arm, and that includes observations from 2012 up to 2020. And we did actually have a few reports in 2021, so we'll need to update this as well. And as I said, this is something that we're seeing both on the US and on the Canadian side. This slide is showing actual photos of blooms that occurred near the Apostle Islands on the south shore and the western arm of Lake Superior. And again, the recent bloom reports have been from 2012 up to present. After 2012, other blooms had popped up along the south shore, and since it can be hard to picture a Lake Superior bloom, I thought I would include some photos. And they can manifest in different ways. These are not necessarily going to be the same as the blooms that we see in Lake Erie or the other Great Lakes. The waters can be green and cloudy with a lot of the material suspended in the water column. Sometimes there are visible surface scums. We've observed these fresh during an event, and sometimes we've observed what looks like older or senescing bloom material. And in any case, while they are happening, the blooms look like blooms you might see in other lakes, maybe particularly in smaller inland lakes. The species of cyanobacteria that's been implicated in these blooms is Dilacospermum lemmermanii, previously known as Anabaena, and as I've heard recently, maybe going back to Anabaena. Dilacospermum has been documented to produce multiple toxins that act on nerves, liver, and other organs. Fortunately, to date, we haven't detected these toxins in samples from Lake Superior blooms. However, toxin sampling has been fairly limited to date, and our colleague Todd Miller at UW-Milwaukee has detected another molecule, anabaenopeptins, in Lake Superior bloom samples. And these could act similarly to algal liver toxins and have effects on aquatic organisms. So what do we know about why these blooms are occurring in Lake Superior? Lake Superior is a cold oligotrophic lake, which is not something that we typically associate with cyanobacterial blooms. And so we are still early in the research, but we do have some evidence that points primarily to climate drivers. For many other lakes, land use is a really important driver of cyanobacterial blooms, particularly in agriculture and urbanized areas. However, in Lake Superior, land use change hasn't really changed much, and over 70% of the land cover in the Lake Superior watershed is still forested, and so this is not likely to be an important driver of cyanobacterial blooms in Lake Superior in particular. While land use hasn't changed much, the temperature in the lake is changing. Anybody who has swam in Lake Superior knows that the lake is certainly cold. However, the rate at which it's warming is very rapid. They found in the O'Reilly et al. 2015 paper, the graphic shown here, that Lake Superior is one of the most rapidly warming lakes in the world by peak summer surface temperature. Additionally, if we look at buoy data from Lake Superior, we find that in these warm years, that not only is the peak summer surface temperature higher, but also the rate at which the lake is warming is faster, which has implication for thermal structuring throughout the stratified season. And so these conditions might be more suitable for cyanobacterial blooms in Lake Superior now and going into the future. But temperature is only part of the story when it comes to climate and cyanobacterial blooms in Lake Superior. We've also seen an increase in the frequency of these really high intensity storm events, and you can see some of the damage shown in these pictures here from the 2018 event. There was also an event in 2012, and coincidentally 2012 and 2018 were the years with the largest cyanobacterial blooms in the western arm on the south shore of Lake Superior. Experimental work has also shown that rivers might play an important role not just for delivering nutrients but also for delivering biological material or seed populations for cyanobacterial blooms in Lake Superior. This slide shows some work from my PhD where I investigated different sources of blooms to Lake Superior to see whether blooms were originating in the lake or in an upland location. And so water samples were collected from in the Lake Superior or lake, the first column in the graph, near the Twin Ports Harbor, as well as from the Siskiwit and Bois Brule rivers. Those samples were collected and brought back to the lab and exposed to high NDP and low NDP nutrient conditions, as well as 15, 20, and 25 degrees Celsius. And what we found is that cyanobacterial growth did not occur under any of the conditions from samples that were collected from Lake Superior, but did grow in samples that were collected from the harbor and the river, particularly in low nitrogen to phosphorus conditions and higher temperatures, 20 and 25 degrees Celsius. The ID results confirmed what we saw and confirmed that in particular the species that we see in Lake Superior, Dilacospermum lemmermanii, was growing from those samples. Based on the results of the 2017 experiments, I investigated 26 different inland locations in 2018 as potential hotspots for cyanobacterial growth and delivery to Lake Superior. These samples were collected and again brought back to the lab. However, this time they were just exposed to conditions that were best for growth in the previous experiments, so low NDP conditions and 20 degrees Celsius. I also measured a large suite of different chemical and biophysical parameters at each location to see if there were shared characteristics among sites that were potential hotspots. And what we found is that there wasn't any single location where we could point to it and say this is our problem spot that is delivering seed populations or biological material to Lake Superior. What we found is that the sites that were potential sources were actually widespread throughout the study area, but we did find that rivers were more likely to be sources than coastal areas or inland lakes and ponds that were sampled in those experiments. The appearance of these cyanobacterial blooms in Lake Superior has elicited a really extensive and coordinated response from both the research and the management communities. Some of these efforts have included the establishment of nearshore and tributary sites, sampling for nutrients, algal biomass, algal composition, and toxins, deployment of sensors for continuous data collection, both in the nearshore and in rivers, laboratory experiments, like I described in the previous slides, as well as those experiments being replicated at other locations, and sediment cores to determine whether or not these blooms are indeed new or whether they have occurred in the past. This coordination occurs through the Lake Superior Partnership Working Group, or the LAMPS Algal Bloom Work Group. This was formed in 2018. It's a binational coordination that's focused on algal bloom issues, particularly cyanobacterial blooms in Lake Superior, and includes federal, tribal, and state agencies that are working with the science community. This group, and myself in particular, have been really involved in developing a rapid bloom response communication plan that entails both notifying people of blooms in the community as well as within the group, and also developing plans for sampling to better understand why blooms are occurring in Lake Superior. And we also have developed standardized field sampling kits for those that are involved in the rapid bloom response plans. As I said before, the appearance of cyanobacterial blooms in Lake Superior was really surprising to not just the research and management community, but also to the broader public, and really grabbed the attention of a lot of media outlets as well as the International Joint Commission. One of the IJC triannual reports recommendations was specifically for addressing cyanobacterial blooms in Lake Superior. And so in 2021, when the Cooperative Science and Monitoring Initiative, or CSMI, intensive field sampling year for Lake Superior came about, cyanobacterial blooms were a common thread in a lot of the efforts in the nearshore. For those of you who aren't familiar with the Cooperative Science and Monitoring Initiative, it's part of the Science Annex of the Great Lakes Restoration Initiative, or GLRI, and it focuses on one of the Great Lakes each year. And so there's these 5 different steps: the field year, the data analysis, report out, priority setting, and planning. And so in any given year, each one of the Great Lakes is in one of those stages. The priorities for CSMI are set under the Lake-wide Action Management Plans, or the LAMPs, for each lake. And then, as I said, 2021 was the Lake Superior intensive field sampling year. Because there was so much work that was occurring on the South Shore in the Western Arm to address cyanobacterial blooms in Lake Superior, there was a major effort led by myself, Anna Baker at USGS, Tom Hollenhorst at EPA, and Ellen Cooney at Wisconsin DNR to coordinate these efforts so that we could get a more comprehensive view of what was happening on the South Shore of Lake Superior. And so what this map shows is just a lot of those efforts. It's not a fully comprehensive view of all of the CSMI efforts, but it's really just a snapshot of coordinated efforts that occurred on the South Shore of Lake Superior, and this included both CSMI funded and non-CSMI funded efforts. I know I've covered a lot in this presentation, and so I just want to provide a brief summary with some take-home messages. First is that the lake is vulnerable to detrimental change. We often think that Lake Superior is immune to the impacts of climate change or human influence on the watershed just due to its sheer size, but we do find that the lake is vulnerable. Unexpected blooms highlight gaps in our monitoring and knowledge, and while that has been changing over the last couple of years, we still have a long way to go. Evidence is mounting that climate change is an important driver. We find that the lake itself is rapidly warming, and that these large storm events, which are increasing in frequency and intensity, might be important drivers of cyanobacterial blooms in the lake. We also have found that upland systems might be important for seeding blooms in the lake, and that rivers in particular might be important potential sources for cyanobacterial blooms in Lake Superior. And finally, we have a window of opportunity to protect Lake Superior instead of being reactionary, as we have been in the past with the other Great Lakes. And so now we're on the cusp of another field season and wondering what the lake will tell us next. I'd like to take a moment to thank many people who have contributed to this work, but in particular, Brenda LaFrancois, Matt Hudson, and Bob Sterner, who have all presented on these topics and provided some of the material that I presented today. And while I would love to take any questions that anyone has, I probably will not be able to be there in person because I'm currently on maternity leave with my 19-month-old and newborn. So if you do have any questions, please feel free to email me at kreinl, kreinl @wisc.edu with any questions that you might have. Thanks so much.